For decades, the way we've studied the human body has been a bit like trying to understand a city just by looking at it from space. You see the general shape, but you miss all the details. So what happens when we finally get to zoom in? Today, we're diving into a revolutionary technology that's giving us a street-level, high-definition view of life itself. Okay, so take a look at the bottom of this image. That's the old way, bulk sequencing. It gives us, well, one single kind of blurry, averaged out result. It's useful, sure, but it hides all the incredible complexity. Now look at the top. That's single-cell sequencing. All of a sudden, these distinct, colorful groups just pop right out of the noise. We're not just seeing the average anymore. We're finally meeting the individual cells that make up the whole. To really get a handle on this massive shift, we're going to need a simple analogy. And honestly, it's a pretty delicious one. We are moving from a biological smoothie to a fruit salad. So for the longest time, whenever scientists wanted to study a piece of tissue, they basically had to put it in a blender. That's bulk sequencing. You get the average flavor, right? There's banana, strawberry, and maybe some blueberry in there. But you have no idea how many of each there were, or if one of those strawberries was maybe a little funky. But now, with single-cell sequencing, we have a fruit salad. We can actually pick up each individual piece of fruit, study it, and see exactly what it's bringing to the party. And this all points to a really, really important idea: cellular heterogeneity. It's a fancy term for a simple fact. The cells in any given tissue are not all identical clones. Nope. They are a diverse, bustling community, each with different jobs, different messages, and in different states of health. That old smoothie method, it completely missed this incredible diversity. But it turns out that diversity is the key to understanding how our bodies really work. So the big question is, how on earth do we actually do this? How do we go from a solid chunk of tissue to this amazing, detailed cellular fruit salad? Well, it's this fascinating three-step process. Let's break it down. Okay, step 1, isolate and capture. Scientists use these incredible little microfluidic chips, kind of like what you see here, to channel a tissue sample into millions of individual cells. Each single cell gets trapped in its very own tiny droplet of oil. And here's the clever part. That droplet also contains a unique molecular barcode. Think of it like putting every single piece of fruit into its own tiny, perfectly labeled container. All right, next up, we read the code. Inside every single one of those little droplets, the cell is gently popped open. Its active genetic material, that's the RNA, gets captured and tagged with that unique barcode. That RNA tells us exactly what that cell was up to at that very moment, what proteins it was making, what job it was doing. Then all of these tagged genetic recipes are fed into a sequencing machine, which reads the entire instruction manual for every last cell. And last but not least, step 3, making sense of it all. This is where magic really happens. A computer takes this mountain of data and uses those unique barcodes to connect every single genetic recipe back to its original cell. It then acts like a master sorter, grouping cells with similar job descriptions together. And what you get in the end is this absolutely beautiful map, a detailed census showing every different type of cell that was in that original tissue. So just being able to identify all the different cells, that was a total game changer. But of course, science never stops, right? The very next question was obvious. Where were all these different cells located in the first place? So look here. On the left, you've got that cell census we just talked about. It's an amazing list of who's in the tissue. But on the right, whoa, that is the next leap forward. It's called spatial transcriptomics. This doesn't just give us a list, it gives us a map. It shows us the exact location of every single cell. So now we can see not just that different cells exist in the heart, but exactly how they're organized into a living, breathing neighborhood. And this quote just nails it. It really is the difference between having a list of guests at a party and having a photograph of the entire room. I mean, think about it. Knowing who's at the party is useful, sure. But seeing a photo of the whole room showing who's talking to who's hanging out by the snacks, that's the real story. And in biology, just like at a party, your neighbors are everything. Okay, this is all incredibly cool science, but so what? Why does this matter to you and me? Well, because this high-resolution view of our own bodies is completely rewriting the rules of medicine. Well, right now, scientists are building something unbelievable, the Human Spatial Atlas. It's literally like creating a Google Maps for the entire human body. By making a perfect cell-by-cell reference map of what healthy tissue looks like, we finally have something to compare against. It lets us see exactly where and how things start to go wrong in a disease. And from there, the applications are just exploding everywhere. In oncology, we can now hunt down those specific nasty cancer cells that are resistant to drugs. In developmental biology, we can literally watch an embryo build itself, one cellular decision at a time. It's transforming everything from fertility treatments to making better crops. Let's bring this home with a really powerful, personal example. This shows the workflow for a patient with breast cancer. A small tissue sample is taken from their tumor. It goes through single-cell sequencing, and the analysis reveals the whole ecosystem of that tumor. The different cancer cells, the immune cells that are trying to fight it, and all the cells that are supporting it. This detailed map helps doctors predict how that specific cancer will behave and choose the absolute best treatment for that individual person. So, when you boil it all down, what's the big takeaway here? It's simple, really. We've just gotten a massive upgrade to our vision. Think about it like this. We've gone from looking at our own biology in a blurry black and white photograph to watching it in a full-color, ultra-high-definition movie. We can see every character, watch how they interact, and pinpoint the exact moments where health starts to turn into disease. This isn't just a small improvement. It's a completely new way of seeing ourselves. This incredible technology is giving us the tools to ask questions we couldn't have even imagined asking just a few years ago. We finally have the map, and that just leaves us with one absolutely thrilling question. Now that we can explore this amazing new territory, what are we going to discover next?